Okay, so I'm just going to get right into it. I'm going to talk about various projects that we're working on and kind of give an update on how herring gulls are being used in the St. Lawrence as sentinels for debris exposure. And I want to start off that this work is being facilitated by my supervisors, Kyle Elliott from McGill and Raf Lavoie from ACCC. And also, I want to acknowledge the many field assistants that are fundamental to all of these projects, and also my funding and where a lot of the samples were analyzed. So just briefly, as we all know, plastic pollution is a major environmental concern, often compared to as urgent as climate change, where millions of tons are entering our oceans and waterways due to gross mismanagement, and it's really threatening the sustainability of many of our coastal ecosystems. And there's no real plan to stop this exponential increase, and this is causing a lot of interactions with wildlife, and we're going to be focusing obviously today on seabirds. And seabirds can interact with plastics in many ways. We're going to talk about how they ingest plastics today, but they can also be entrapped and cause many different adverse effects. So plastic pollution monitoring in Canada is pretty widespread, and in many ecosystems, long-term monitoring projects have been ongoing, but there are some systems where are kind of being lacking information, and the St. Lawrence is one of them for seabird plastic pollution. And the St. Lawrence system has high biological productivity. It sits downstream from the highly polluted Great Lakes, Laurentian Great Lakes. And there's increasing studies that show anthropogenic debris are being introduced into these systems along with contaminants. Recent evidence shows that the proximity to large industrial centers like Montreal, you find increasing levels of plastics. And also, we know these cumulative anthropogenic stressors are working at the same time, like shipping, transport, fishing intensity. And millions of seabirds are relying on the system as well, not only for breeding, but during molting and wintering, where they really rely to forage and within the system. And this is just a cool video of a colony in the St. Lawrence. So today I'm going to try to convince you that herring gulls are effective monitors for plastics and debris in general. And one is because they are distributed throughout the St. Lawrence, but they're also decreasing since the fishing collapse in the 90s. They didn't really recover their population sizes. So, and we think, and recent evidence has shown that this has maybe urged them to start to look using resources, anthropogenic resources, and fishery discards. Also, they're opportunistic scavengers, as we know, where they can feed exclusively on marine diets, also terrestrial sources, and as we all know, right, garbage. And they can also have mixed strategies, and this is largely based on where their colony is located. So they have this increased threat of plastic consumption, and we can use them to kind of understand environmental pollution, like plastic pollution within a system. And I just included this picture where when you're going through a herring gull colony and nest sites, like it's very common to see plastic waste, right, within nests and around nest structures. So our first question that I'm going to try to be answering is how plastic exposure changes with foraging strategy in seabirds across the St. Lawrence system for the first time. And we used 4 different colonies as shown here with varying proximities to industrial areas, Quebec City and Montreal. So our general question is, will remote colonies have less plastic, and will it vary based on the foraging strategy of the individual? So this is not my project. This is my honors student project where she analyzed Caitlin Goodwin, who analyzed over 600 pellets from these 4 different colonies. This is a large amount of work. And these are opportunistic pellet collections. So we're putting forth these minimally invasive techniques of looking at plastic exposure. So in terms of results, what she found was really interesting. So we found that, and first on the x-axis here, this is the left to right is increasing distance away from Quebec City, the 2 colonies within the estuary and 2 colonies in the Gulf, more remote. And then on the y, this is the percent of pellets that included the debris type. Gray is none, and then plastics are that lavender color. And you can see that we did find increasing plastic debris and actually debris in general closer to Quebec City and in industrial cities. And then Bechuan, the most remote colony, had the lowest amount of plastics. Just to give you some numbers, so 62% of pellets from the closest colony compared to 20% at the farthest. But I also want to stress that plastic was prevalent across all colonies. Okay, and then when we answer the second question, and now this is the same, so it's increasing distance away from Quebec City, but now we split into what the inferred foraging habit, but looking at the diet within the pellet, tell this based on debris type. So we found that, as you can see on the x-axis, there's terrestrial, mixed, and aquatic foraging strategies. And these remote colonies, so the rightmost of the graph, you can see that have these aquatic foraging strategies. And then when you compare it to the colony closest to Quebec City, we had largely these terrestrial foraging strategies. So it makes sense. We see this huge shift in foraging strategy based on location, but we also see this shift in plastic exposure, right? So these aquatic pellets had less plastics. So this really shows that the signals not only about where they're foraging, also the colony location. So these are working hand in hand. And we also, through FTIR, looked at the polymer composition. And what we found, just briefly, is that PE and PET were dominated all colonies. That's the pink and magenta colors here. And these are single-use plastics, right? So it's like garbage that you would find in human trash. But then on this much more remote colony, it was dominated by PVC, which is more like construction type plastics and things like that. So we actually find this fingerprint of like plastic type and characteristic differences based on colony location. And just to go to my PhD, which focuses on plastic-related contaminants, we kind of tried to answer the same question: how colony varies contaminant burdens, and we found the same pattern that we find with plastic itself, is that these plastic-related contaminants, so the plastics that leach out of the plastics, were correlated with distance from Montreal, and we found higher concentrations closer to urban areas. Okay, so now that we got that out of the way, we want to know also, I'm really interested in foraging ecology. So my next question is, what is driving this variation? So we know plastic ingestion is widespread, but on the individual level, how does foraging habitat shape ingestion? And does behavior at the individual level predict exposure? So we're going to go to these industrialized locations, and just taking you back to this colony here, which is the Bellechasse colony. This is a colony that's considered the most industrialized out of these. It's about 20 kilometers away from Quebec City, second most industrialized city in the St. Lawrence. And what we did is we did this two-pronged approach. So we collected pellets over 2 breeding seasons, and we also GPS tracked adults from the same nest that we collected pellets from. And on the pellet side, we looked at the occurrence, the count, and the mass, and the type of debris. And then we paired this with the GPS tracks and wanted to understand how site fidelity, landfill use, so that using waste management sites and their home range. So how does this foraging behavior maybe impact the debris exposure found in the pellets? So for results, we found that 47% of all pellets contain debris, and 40% contain debris, so plastic and not plastic debris, and 40% of all pellets contain plastic. So a lot of it was plastics. And diet was interesting as well. So 64% of the diet was anthropogenic diet items. So items you would find in a landfill urban area waste, compared to 90% were agricultural, so grains, terrestrial insects, and 17% were only aquatic. And 24% were terrestrial animals. And we also found that these patterns were similar across years. So we have this chronic exposure, not episodic, right? So this is chronic plastic exposure. Going back, we did FTIR analysis again on the polymers, and I think this is pretty interesting that in these anthropogenic diets, you find these single-use plastics, like PET and PE. But then when we look at the only aquatic, though they were rare, the pellets that were only aquatic were rare, we did see this shift, and it was polystyrene, so PS polystyrene, which is very buoyant plastics, foam-like plastics, marine foam plastics, so maybe plastics that are floating on the sea, the St. Lawrence surface, and that's maybe how they're foraging on these. So this is interesting. Again, we see this difference in polymer composition, like we saw in the large scale spatial assessment project. So these plastic fingerprints based on habitat. So in terms of anthropogenic pellets, I just want to give you some results here. The increase, so pellets that were deemed anthropogenic, so feeding on garbage-like resources, had higher probabilities of plastic occurrence, higher counts of plastic, and actually heavier plastics, which is like, it's what we assume, but it's cool to see these results, right? And then these agricultural and aquatic pellets, well, they still had plastic occurrence. They had lower debris loads and lower debris loads, lower plastic item counts, and lower masses. So it confirms that debris ingestion is not just random, it's strongly linked to your foraging patterns. And this is just to show you the GPS results. In green is the agricultural foraging, and I just want to stress how important agricultural foraging we're finding is in these coastal systems. And 56% of individuals, 100% of individuals foraged in an agricultural field at least once, and 56% of individuals foraged in a waste management facility in the track period. But when we look at landfill use, whether an individual just exploited a landfill or it didn't, it was actually a weak predictor of plastic ingestion. So they are uptaking these plastics within these agricultural fields. But we did find, interestingly, that site fidelity, so more exploratory behavior, actually caused individuals to ingest more plastic than consistent foragers. So increasing site fidelity, the less places you're going to explore, we found that actually predicted lower plastic counts and lower probability of plastic being in the diet. So from this suggests that foraging behavior can influence the exposure to this debris. And going back another chapter of my PhD, we did find the same pattern. So individuals that visited a waste management facility did not predict whether an individual had higher levels of plastic-related contaminants. So we're seeing these, despite these landfills and waste management facilities being major sources of plastics and their contaminants, we're not seeing these patterns. So it suggests that agricultural fields are contributing to exposure. So my take-home message quickly is that debris ingestion is widespread, but it seems to be structured. And we found my major points is that anthropogenic or terrestrial foraging really drives this plastic ingestion, and exploratory behavior, not whether an individual simply exploits a landfill, increases exposure risk. And doing these integrative assessments with pellets and movement improves our precision on kind of understanding at the individual level how foraging ecology may influence contaminant burden. And that's all I have. Thank you. Sorry, that was fast. I had a lot to go through. I'll take any questions though.
Yeah, you have about 2 minutes for questions.
Yeah.
I have a quick question, actually.
Sure.
I am wondering about, so you had mentioned that your further away colonies, there's more marine plastics that would occur in the marine environment. So are you assuming that that these gulls are actually picking the plastic up from the marine environment, like from the ocean, or are they? Do you have any idea about where the source is coming from?
That's a good question. We didn't specifically. You're saying like, was it in a fish? Yeah.
Yeah, or even just
not.
are they getting it from land as well, and then finding it?
Yeah, they could be. Definitely an assumption is that we just generalize the foraging habitat, the diet inferred from the pellet is how they were getting that plastic. But yes, they could be getting that just from land as well. That's an assumption that we're doing with the pellets.
Right.
Oh, I
I think
have
we.
quick.
Oh yeah, you
Okay.
have one more question here, and then I think after that we better move on.
Okay, perfect. Quick question about anthropogenic foraging. Do you have any sense that those anthropogenic foragers produce more or less chicks per year? Are those more urban colonies growing, decreasing? That's a great question, and this is a future research that we're hoping to come out of these kind of projects. We haven't looked at these yet. This is we started these projects in 2022, so and we're trying to keep them going. But that's a great question, something I'm really interested in. And I can
Um,
answer any other ones in the Q&A after.
yeah, that sounds great. So if Erin can come
So,
on, and yeah,
yeah.
sorry, I'll let you.
Yeah, so next we're going to be sticking on the plastic route, but with Erin Murphy, who's going to be talking about, oh, Erin, are you here?
I'm here, yeah. I'm
Okay,
working on
perfect.
sharing my slides as
Okay,
we talk.
perfect. I just make sure.
I'm also a Zoom person. And yeah, I live in Alaska, so it's before 7, so I need y'all to bear with me as I wake up.
Good
Thanks for
morning.
joining us so early, but you did a great job. We can see it. Looks great.
Awesome. I'm very happy to be here.
Okay, so take it away, Erin. Thanks.
Yeah, hi everyone. So my name's Erin Murphy, and I'm the manager of ocean plastics research at Ocean Conservancy. And today I'm going to be talking about a risk assessment framework for mortality due to macroplastic ingestion in seabirds. This is a subset of data from a recent publication that we have, and I'll have a QR code so you can see the publication at the end that focuses on seabirds, marine mammals, and sea turtles. But I just wanted to share these specific seabird results with you and also talk about what we're hoping to do next with this body of work and see if there's any opportunities to work with folks that are doing seabird and plastic work. So every family of seabird has been documented to ingest plastic, and I don't need to necessarily continue on after that last presentation about plastic exposure through ingestion in seabirds, but macroplastic ingestion can cause injury, illness, and death. And this is something that we've documented in all, or in these seabird families. But the question that really remains research-wise is how much plastic is too much. So, we observe these instances of death or injury in seabirds, but we don't necessarily know the relationship between what they're ingesting or that gastrointestinal load and then their likelihood of mortality from plastic ingestion. So the goal of this larger research program at Ocean Conservancy is to develop a suite of monitoring and management tools for macroplastic so we can better track this problem, identify risk thresholds, and inform guidelines, goals, and regulations. And so far, this research kind of focuses some on that risk assessment methodology. We've also been working on a monitoring protocol, and then our ultimate goals are to develop our comprehensive risk assessment methodology and a risk management framework. So just giving a quick overview of risk assessments, this is just a process to identify potential hazards, and there's two components of risk assessments. The first is exposure. So what amount of contaminant is an animal actually being exposed to? And then the second piece is severity of harm. So if this exposure occurs, how likely is harm to occur? And these can be quantitative, qualitative, semi-quantitative, or scenario-based. And there's been a lot of work on microplastic risk assessments. And these are a little bit easier to conduct because you can focus on the impact piece in particular in the lab. And so what we wanted to do was to develop impact assessment methodologies to inform risk assessments for macroplastic ingestion, which are harder to study in laboratory settings or unethical to study in laboratory settings. And so specifically, our research goals were to quantify the relationship between plastic that's in the gut and then chance of death. And we talked specifically about acute mortality here. So we're working with, we conducted a literature working with animals that have died and are being assessed during necropsy. And so we focused on acute mortality where we see clear obstruction of the GI tract, puncturing of the gastrointestinal wall, or torsion of the intestines. So it's clear that that death occurred due to the plastic in the gut. And ultimately, we want to develop these models that you see on the right, where you just see the increasing amount of plastic on the x-axis and the likelihood of mortality from plastic ingestion on the y-axis. And we also wanted to explore lethality of different plastic types. And so we collected data, not just on total plastic ingested in these animals, but also on the different plastic types. And for seabirds, we had enough data to also look at hard plastic fragments separately and rubber, synthetic rubbers like balloons. And then additionally, we wanted to summarize research on ocean macroplastic ingestion and mortality. So there's a ton of data out there on what these animals are eating, and we just wanted to get an idea in the literature of ingestion rates and mortality rates that are reported. So our approach to develop this model was to first build a global database. So we reviewed the literature on macroplastic ingestion, and we extracted necropsy data. We focused on necropsies and not pellets or lavage or feces, just because we needed to have this evidence of acute mortality. And then we developed a model to look at this relationship between plastic in the gut, so gastrointestinal load at one snapshot moment, and likelihood of mortality. And we piloted this on seabirds. Like I said, we also looked at marine mammals and sea turtles if folks are interested in that. And then we identified global thresholds for this different taxa and species as well, and published this open access so others could use this tool. We looked through the literature, just, from 1900 to 2023, covering anything that we saw. And our search terms, you can see, are ingestion, plastic debris, marine debris, macroplastic, mesoplastic, and fisheries debris. We included both peer-reviewed literature and stranding network databases from the United States, which is just where we had access to those. And to be included in this model, we had to have one of these three taxa. It had to focus on macroplastics greater than 5 millimeters, and the cause of mortality needed to be reported. So we didn't necessarily need to know the exact cause of mortality, but we for sure needed to know if plastic ingestion was the cause of mortality. And I'll just note that we did include studies here where, every animal was bicaught, for instance, or had a fishing gear interaction, and none of the mortality events were from plastic ingestion because we didn't want to overinflate, but we just needed to know why these animals died. And we also needed high resolution data on the ingested plastic. So not just the total pieces of plastic ingested, also the volume of plastic ingested, and then the number and volume of plastic ingested by plastic type, which also included hard plastics, soft plastics, fishing debris, foam, rubber. So even the things we are unable to model in terms of plastic type, we still have data on ingestion. And then for data collection, we collected organismal data, family, species, biological traits that were available about the animal, and then the location and collection method and the cause of death. And in terms of plastic, like I said, we collected data on each plastic type and on the number of pieces and the volume. And then if plastic was the cause of death, we specifically noted what piece of plastic caused death. Was it, a grouping of plastics that caused an obstruction, or did we see perforation from a specific material or obstruction from a specific material? So here's just a summary of the necropsy data that we had for seabirds. So we had more than 1,500 individuals, and 65% of seabirds ingested no plastic. From these studies, 35% did. And of the 35% that did, 5% died of plastic ingestion and 95% did not. So based on this, we modeled the relationship between the number of pieces in the GI tract and the chance of mortality for plastics generally. And we found that 12 pieces of plastic ingested at one moment, right? So this is their gastrointestinal load at a given time, was associated with a 50% chance of mortality, and 28 pieces was associated with a 90% chance of mortality. And then on the right, the way that we modeled volume in its relationship with mortality was